solute imbalance. So it's kind of like one of those bath time squeeze toys where you squeeze it and water comes out. So it gets the water out of the cell. The contractile vacuole enlarges as water enters, then quickly contracts to force water out through the special pores. Then we have the cytoskeleton, which is the cell's skeleton. This supports and maintains cell shape. It holds organelles in position. It moves organelles. It's involved in cytoplasmic streaming. It interacts with extracellular structures to anchor cells in place. So it has a lot of uses. So they can be between cells to hold them in place. They can keep them tight. They can let stuff go through, or they can be super tight, like stitches along the edge. They're also inside the cells, keeping them together and in a certain shape. And they're also outside of it as well, protecting it. So in your blood, for example, you have a lot of flat cells surrounding your blood going through. So there's your blood going through. But you also have an extracellular matrix that protects your blood vessels from the stuff in the blood. So it protects from LDL and things like that from sticking to them, which is super useful. So yay, extracellular matrix. So the cytoskeleton has 3 components with very different functions: microfilaments, intermediate filaments, and microtubules. So the first one, microfilaments. They help a cell or parts of a cell to move. They determine cell shape, and they're made from actin monomers. So this is a type of protein that attach to the plus end and detach at the minus end of the filament. So you have your actin, and it makes a filament. And you have your minus end and your plus end, and new ones attach over here, and some can detach over here. So this helps it grow and get smaller where it needs to. So these are each actin monomers. So here's a better picture of that. It's about 7 nanometers wide. There's the plus end and the minus end, and it's made up of actin monomers. These are used in cells. Here's the cell membrane, here's the nucleus, and here are all of the filaments. These are microfilaments. They're very small, 7 nanometers. So they have dynamic instability. The filaments can either shorten, more detachment, or lengthen, more assembly. So as I said before, how they have the minus end and the plus end. So things can add to the plus end to grow or make things spread apart, or they can detach from this end to shrink and let things come back together. This allows for quick assembly or breakdown of the cytoskeleton. In muscle cells, actin filaments are associated with the motor protein myosin. Their interactions result in muscle contraction. Motor protein, or the molecular motor, any protein that causes movement within a cell. So this can cause movement, so they can grow to make a part of the cell go out, or they can shrink to let it come back in. So there are intermediate filaments. There are at least 50 different kinds in 6 molecular classes. They are tough, rope-like protein assemblages. They are more permanent than other filaments and do not show dynamic instability. So do not show that. So they're pretty much there, and they're kind of like trying to think of a good. So intermediate filaments are kind of like wood that's for building houses, and the microfilaments are kind of like scaffolding. They build it up when they need it and they take it down when they don't. So micro equals scaffolding. Intermediate is like house building. So the wooden house building is going to be there. It's not going to change size. That's how it is. So they anchor cell structures in place, they resist tension, and they maintain rigidity. So here is the fibrous subunits. So this is a lot bigger, 8 to 12 nanometers. So they have these subunits. So in each of these, so you kind of have an end that looks like this wrapped around another end that looks like that. So there's multiple ones there. There's 2 types wrapped around each other, and they're each made of smaller subunits. So then we have our microtubules. These are the thickest cytoskeleton elements. They form a rigid internal skeleton for some cells or regions. They act as a framework for motor proteins to move structures in the cell. So these are like your steel beams. So your microtubules are like your steel beams where you're building your house, you put up a couple steel beams to support the big, nice, open concept people like. And then you have your wood to make the floors and things like that. And then you have like scaffolding around it to maybe install a window. So these are microtubules. These are intermediate filaments. And then these are microfilaments. And these are your microtubules. So we have our microfilaments, our intermediate filaments, and then our microtubules. So microtubules have 2 subunits. They have beta tubulin and alpha tubulin. And they make a dimer called tubulin, made up of the beta and alpha. Because remember, you have monomers, dimers, and then polymers for anything 3 or above. So here's the structure of microtubules. They're tube-like. and they are made up of these dimers of beta alpha. And they are like the steel beams that hold up the cell. They're 25 nanometers wide. Microtubules are made from dimers of the protein tubulin. Chains of dimers surround a hollow core. They have a positive and a minus end and show dynamic instability. So they can shrink or grow if they need to. Polymerization results in a rigid structure. Depolymerization leads to its collapse. So remember, polymerization is just them coming together, and depolymerization is them breaking apart. Microtubules form an internal skeleton from movable cellular appendages. So they form the cilia and flagella. Cilia are short, usually many present. They move stiffly to propel a cell or move fluid over a stationary cell. So think of them as like paddles on a ship or a rowboat. So the cilia, they move stiffly along to propel the cell forward. Flagella are longer. They're more like a engine in the back of a boat to propel it forward. So they rotate to propel it forward. So they push or pull a cell through water. So here are the cilia. They're short, they move kind of like oars. And then here is that microtubule. If you do a cross section of that, this is what it looks like. You can see the dimers in it and how they build up and have a central one with motor proteins in between. So these are each microtubules, and they come together to make the flagella, or the cilia. Okay, so cilia and flagella microtubules are arranged in a 9+2 pattern. They have doublets. 9 fused pairs of microtubules form a cylinder, and then there's one unfused pair in the center. So they have the ones around the ring, and then the one in the center. Motion occurs as doublets slide past each other. So these tubules, some can be up and some can be down, and this causes it to bend in the direction that the longer one is. So dynein is a motor protein that can change shape and drives the sliding of doublets. So it's normally in between them. Nexin is a protein that cross-links doublets and prevents sliding. So cilia bend instead of just going past each other. So it keeps them together so that they actually bend when that happens. Kinesin is a motor protein that binds to vesicles in the cell and walks them along the microtubule. So it kind of just looks like a dude walking along a microtubule. So that's kinesin for you. So here is the dynein. It moves them up and down. Here's the microtubule doublet. Here's an exon. It keeps them bound, kind of like a ladder rope. That's thionine. So when these cause it to move, it bends. So here is the microtubule and the kinesin. You can see how it walks along it. So the one that was in back comes forward, and then they just keep walking down the tubule like that. So cytoskeletal structure may be observed under the microscope, and function can be observed in a cell with that structure. Observations may suggest a structure has a function, but correlation does not establish cause and effect. This is big in science. So two methods are used to determine a link between a structure A and its function B. Inhibition, you use a drug to inhibit A, and if B still occurs, then A does not cause B. There's also mutation. If genes for A are missing and B does not occur, A probably does cause B. So here they did an investigation to see how amoebas move, or amoeboid type cells move, and to see if they're caused by the microfilaments. So here we have Amoeba proteus. It's a single-celled eukaryote that moves by extending its cell membrane. So the amoeba is treated with cytochalasin B, and this one is control injected, but without the drug, to see if just poking with a needle makes it stop moving. Cytochalasin B is a drug that blocks the formation of microfilaments. Really bad news for a cell. So the treated amoeba is alive, but runs up and does not move because its microfilaments are just dots around the cell and can't do anything. So the control amoeba continues to move because it has lots of nice microfilaments that allow it to extend or contract as needed. So the microfilaments are essential for amoeboid cell movement. That's how they determined cause and effect. So extracellular structures are secreted to the outside of the cell membrane. In eukaryotes, these structures have two components, a fibrous macromolecule and a gel-like medium in which fibers are embedded. Do I want to say anything more about that? Not really, it's just kind of the same model of secretion there, where it's embedded in the vacuole, it fuses, and then it's now on the outside of the cell. And that has this gel-like stuff where the fibers are all embedded. So the plant cell wall is a semi-rigid structure outside the cell membrane. The fibrous component is the polysaccharide cellulose, aka fiber. Gotta have that cellulose. The gel-like matrix contains cross-linked polysaccharides and proteins. So the cell wall is made up of protein and the sugar building blocks. Proteins in between. And there's some gel-like stuff in between because it absorbs the liquid. So here you have the cell membrane, and then you have the cell wall here. Here's another cell wall and another cell membrane, and here's another cell wall and another cell membrane. Then here's the middle lamella where they don't quite come together. The plant cell wall has three major roles. It provides support for the cell and limits its volume by remaining rigid. It acts as a barrier to infection. It contributes to form during growth and development. Adjacent plant cells are connected by cell membrane link channels called plasmodesmata. These are kind of like gap junctions in animals. So it's basically just two membranes or cell walls that have holes that go through. That's what a plasmodesmata or a gap junction is. They allow things to go through quickly without having to go through the cell membrane or the cell wall. These channels allow movement of water, ions, small molecules, hormones, and some RNA and proteins. Many animal cells are surrounded by an extracellular matrix. The fibrous component is the protein collagen. So this is what gives your skin nice and bouncy. That's collagen for you. It's a protein. The gel-like matrix consists of proteoglycans. So that's protein and glycan. So it's a protein, carbohydrate. A third group of proteins links the collagen and the matrix together. So you have your collagen and your tubules and your proteins along the surface, and then there's proteins in this as well floating around, and then you have connectors. So here we have our kidney cell. If we look closely, here in the electron microscope scan, you can see the proteoglycan with its very changed structure, with its proteins and its carbohydrates, carbs, proteins linked together, and it keeps the collagen linked together. So the extracellular matrices in animal cells hold cells together in tissues, contribute to physical properties of cartilage, skin, bone, and other tissues. They help filter materials, for example in the kidneys, and they orient cell movement during development and tissue repair. Proteins such as integrin connect the extracellular matrix to the cell membrane. So remember when I drew the cell membrane and a protein and the connector, so this would be integrin. And then you'd have the matrix on top of that. These proteins bind to microfilaments in the cytoplasm, and so there's microfilaments down here in the cytoplasm, so they bind it all together so that it's not connected just to the semi-fluid membrane. For cell movement, the protein changes shape and detaches from the collagen. So it can let go and connect over here if it wants. So there's the outside of the cell, the extracellular matrix, and then you have actin and your microfilaments inside. Here you have your proteins, which are the integrin, And then you have your collagen fibers up there. They can let go and allow movement of the extracellular matrix. Cell junctions are specialized structures that protrude from adjacent cells and glue them together. I mentioned gap junctions earlier. There are also desmosomes and tight junctions. We'll go over each of those in the next couple slides. So tight junctions prevent substances from moving through spaces between the cells. So these are kind of like the sewing stitches. They keep it nice and on lock, and nothing can go back and forth. The desmosomes hold cells together but allow materials to move through the matrix. So these have room. And then the gap junctions are channels that run between membrane pores in adjacent cells, allowing substances to pass between the cells. So these are more like tubes that go through. So here we have our tight junctions, like sewing stitches. Desmosomes. This is more like a rivet. It keeps it together but allows stuff to go through. And then gap junctions are just tubes, which still keep it together. But stuff can go through here, stuff can go through here, but it gets stopped there. It's a wall. And this is the basolateral region, which you don't need to remember. So here we have our tight junctions. You can see they look like sewing stitches keeping quilting together. Here's an electron micrograph of them. The proteins of tight junctions form a quilted seal, barring the movement of dissolved materials through the space between epithelial cells. Epithelial is just a tissue type. It's mainly skin, intestines, vessel walls. That's epithelial for you. And then here we have our desmosomes. They link adjacent cells tightly but permit materials to move around them in the intercellular space. Here's an electron micrograph of that right here. Here are keratin fibers, which are cytoskeleton filaments, and they interlock together kind of like Velcro. And they allow things to go through. And then you have your gap junctions, which allows cells to communicate quickly. You can see those here, and stuff can go through. So here's the answer to the opening question. Synthetic cell models, or protocells, can demonstrate how cell properties may have originated. Combinations of molecules can produce a cell-like structure with a lipid membrane and water-filled interior. As in modern cells, the membrane allows only certain things to pass, while RNA inside the cell can replicate itself. Here is a picture of a protocell. You can see the RNA and the cell membrane, and then the nucleotides floating around that helped make it up. So they replicated early conditions so that these would form. Life probably began with a single-celled organism resembling modern bacteria. These cells lack structures that are typically preserved as fossils, but recent advances in microscopy have shown 800 million year old fossil cells that resemble the protocells. And that is it for chapter 4. Feel free to rewind, watch it again, whatever you need to do to remember it.